Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 18th video in the series of videos on immunology. And this particular video is the part 2 on the pathogen recognition molecules. In the part 1, I've told you that pathogens, they express a few chemical structures that are not typically found in mammals. And these structures are known as the pathogen-associated molecular patterns, or they are also sometimes known as the microbe-associated molecular patterns. And I give you many examples of the pathogen-associated molecular pattern like flagellin, the lipopolysaccharide, the peptidoglycan, the zymosin, and the nucleic acid of bacteria and viruses, the coat protein of the viruses. There are different anchors on the parasites. So these were the important examples from the pathogen-associated molecular patterns. Then I've told you that to come in contact with these PAMPs, the immune cells, they express receptors on their surfaces, which are collectively known as the pattern recognition receptors. And these pattern recognition receptors, they are capable of recognizing the PMPs, thereby neutralizing the pathogens. The PRRs, I've told you, they are divided into 4 families. One is known as the toll-like receptors. The other one is known as the NNR. The third one is the CLR. And the fourth one is the RLR. And the interaction of the PRRs with these PMPs, that is going to lead to a cascade of events that label the target pathogen for destruction. Then I've told you that this interaction is very important because it is responsible for the profiling of the sort of pathogen of concern and directing the subsequent immune response. I give you that if you talk about the viruses, that viruses, they are intracellular receptors, and for them the viruses, they are detected by the intracellular receptors inside the host cells, and that particular interaction leads to an antiviral response, and at the same time that is going to activate a neighborhood watch system. Now in this particular video, I want to focus on that the host pathogen interactions, they are an ongoing arm race. What I mean by that is that the pathogen evolve themselves to express unique structures. By that I mean they are going to evolve to express unique PMPs that avoid the host detection system, thereby enabling them to increase their population. Host on the other side, they are going to co-evolve, and the host, they you can say they have got these germline-encoded recognition system in the form of the PRRs. So they co-evolve to match these new challenges. So the PMPs, they evolve, the PRRs, they evolve so that the host can recognize the pathogens. The problem is that if you talk about these pathogens, they generally have like a much shorter life cycles than their vertebrate hosts. Say for example, if you talk about the bacteria and the human beings, so the bacteria, that is going to have a very shorter life cycles, and some utilize error-prone DNA polymerase to replicate their genomes. What this means is that the pathogen, they evolve rapidly to evade the host-encoded recognition system. So is the evolution is very much quicker in these pathogens because of their shorter life cycles. They evolve much quickly as compared to their host. Now, if this were our only defense, what I mean by this is that if we have fixed PRRs, that means that our host immune response, that would quickly become obsolete. If the pathogen is evolving, if the host is not evolving, there would be a time that our immune system will not be able to recognize these pathogens. This gives rise to like a sort of questions, like how the immune system prepare for this. If the pathogen that is evolving, how the immune system is preparing itself to co-evolve and in a manner to you can say counter the new PMPs produced by these pathogens. Secondly, how our DNA encode a recognition system for this thing that change in random ways over time. How our DNA, that is going to evolve itself to come in contact with the new PMPs produced by these pathogens in the process of evolution. Thirdly, how do we build a system to recognize new chemical structures that may arise in the future? These are some of the basic questions. How the host is going to adapt itself to the evolution in the pathogens. If you talk about the human being specifically, for example, if you talk about the vertebrates, So the vertebrate immune system has evolved a very clever strategy about this evolution of the pathogens. And this particular strategy is known as the generation of diversity. And this generation of diversity is employed only by the developing B and T cells. We are not talking about the mature B and T cells. This generation of diversity is employed by the developing B and T cells, lymphocytes. Now, due to this generation of diversity, there is a group of B and T cells where each express many copies of one unique recognition molecules. We are talking about the PRR over here. You should keep in mind this thing, that one B cell is going to express a unique recognition molecule. So the other B cell that is going to produce a PER, that would be totally different than the PER produced by the first B cells. If you have got two B cells, you are going to have two PRRs. What this means is that every B cell is going to express a unique recognition molecule. Now, this results in a population with the theoretical potential to respond to any antigen that may come along, because you are going to get, you can say, a lot of B and T cells with different recognition molecules on their surfaces. Now, this feat is accomplished by rearranging and editing the genomic DNA that encodes the antigen receptor expressed by each B or T cells. The number of genes for these receptors, that is limited. What that means is that you have to rearrange the DNA, you have to edit the genomic DNA so that you can express a lot of PRRs on specific B and specific T cells. Now, not unlike the error-prone DNA replication that I've told you that is employed by the pathogens, this system allows a chance to play a role in generating a menu of responding recognition molecule. So a lot of PRRs, they are produced on the developing B and T cells, and that can theoretically attach to all of the pathogen associated molecular patterns that are produced by the pathogens. Now, this cutting and splicing of chromosome is of course not without a risk. So if you are editing the DNA, if you are rearranging the DNA, risk is associated with this. And many B and T cells, they do not survive this DNA surgery, all of which take place in the primary lymphoid organs. For example, if you talk about the T cells, so the in the thymus, this DNA rearrangement is going to take place, and if you talk about the B cells, this DNA rearrangement is going to take place in the bone marrow cells. What this means is that some of the cells, they are going to survive, some of the cells that are not going to survive. So the surviving cells, only the surviving cells, they move into the circulation of the body, where they are available if there is specific antigen, and they are going to encounter that particular specific antigen which have specific PAMPs. Now, when antigen bind to the surface receptor on these cells, that is going to trigger the clonal selection. Please refer to my earlier videos on the clonal selection, where I have a detailed discussion on what this clonal selection means. So what this means is you are going to edit the DNA of the B and the T cells when they are developing. Only the surviving cells, they are going to move into the circulation, and they are going to encounter the PMPs available on the surface of the pathogens, thereby leading to the clonal selection of the specific B and T cells. Now, the selected clone of the B cells, that is going to create an army of the cells, all with the same receptor, and they are responsible for binding more of the same antigen with the ultimate goal of destroying the pathogen in question. So, for example, if you have got 10 B cells, out of these 10 B cells, one B cell recognizes a specific PMP. So, the population of that particular B cell is going to increase. A lot of, you can see, those particular B cells, they are going to be produced, and those particular B cells are going to neutralize the antigen in question, or neutralize the pathogen in question. Now, in B lymphocytes, like these recognition molecules, most of the time they are the B cell receptors. When they are present on the surface of the B cells, we call them as the B cell receptor. But in some cases, these recognition molecules, they are in their secreted form, and in their secreted form, they are known as the antibodies. When you talk about the T cells, there is no soluble form of these recognition molecules. Therefore, they are called as the T cell receptors. Now, this is, you can say, a very amazing discovery made by the immunologist. And in 1976, the Suzumu Tonigawa. You can hear a difficult name. This particular scientist it discovered the molecular mechanism behind the DNA recombination events that generate these B cell receptors and antibodies. And for this astonishing and for this you can say important discovery, he was awarded the Nobel Prize in Physiology or Medicine in 1987. So if you like the video, please subscribe to my channel, hit the notification bell, do comment on the video. If you have any question, don't hesitate to ask me, and do share it with your friends. I'll see you in the next video.